I'm Sai Chavala. I'm the co-founder and chief scientific officer of CiRC Biosciences. CiRC Biosciences is a cell therapy company, and we believe with a chemical cocktail, we can take an ordinary skin cell and turn it into any cell type in the human body. And those cells can be used to restore vision, it can be used to improve heart function, may be able to be used to help with diabetes. So we believe that the chemical reprogramming approach allows us to turn ordinary skin cells into cells that would improve disease and improve function. And cell therapy has evolved, so you may have heard about stem cells, which are really exciting, but unfortunately with stem cells there's some dangers that come with that. And they can form tumors, and also they can become any cell type. But because they can become any cell type, when you transplant them into a diseased organ like the eye, you don't know if the cell type that you put in there will maintain that same cell identity. So at CiRC, we've come up with a chemical reprogramming approach. So we can take small molecules, drugs that we normally have, we can put them in a culture dish and transform ordinary cells that are abundant, like skin cells, into other cell types that may be useful to restore vision, in our case, or other diseases. So we think in about 2 to 3 years, we'll be able to satisfy the requirements by the FDA to show that our cells are safe in animals, and then be able to advance that to a clinical research trial where we'll hopefully enroll patients that are completely blind, have very little to lose, and be able to demonstrate that, one, the cells are safe when they're injected in these individuals, and that they can actually improve vision. So retinitis pigmentosa is the most common form of genetic blindness. So this is a disease that is inherited from either the mother or the father. And if there's a gene that's mutated or is irregular that causes the cells of the retina to die. And over time, years and years and years, these patients first lose peripheral vision and then they lose their central vision to the point where they're completely blind. The problem with retinitis pigmentosa and other inherited retinal degenerations is that there's over 90 to 100 genetic mutations that are completely different that cause them. So it's very difficult to imagine one therapy that could cure them all. But cell therapy has that potential benefit. We could take cells from a healthy donor and potentially transplant them in the retina of someone who's blind, and we could potentially restore vision because the healthy donor cells will not harbor that same genetic mutation as the patient carries. Retinitis pigmentosa has a huge unmet need, just like dry macular degeneration as well. But we think that if we use a healthy donor, we could first target retinitis pigmentosa. And once we show that, we could potentially use the same cell type or an individual cell for dry macular degeneration. I'm really focused on treating patients and finding new therapies to be able to benefit my patients. And that's why I got into science. So science I felt was like this new language that I could learn. And if I understood the language, then I could potentially bring new therapies to conditions like dry macular degeneration or retinitis pigmentosa, where there are no therapies. And so once we made this breakthrough in my research lab, we realized that there was a whole nother aspect of bringing this kind of therapy from my lab to a patient. And that's where Paragon came in. Paragon allowed me to be able to focus on the science and focus on what matters to me, while the other aspects of drug development were tackled by a highly experienced team of individuals that had done this before, and allowed me to kind of create new therapies or refine our technology and just focus on the science.